Hello friends, how are you? Welcome back to my channel. So today in this video we are going to see diversity in living organisms and about different types of cells which are included in the textbook. Okay, so we will conclude this cell, the basic unit of life, in this video.
Hello friends, let's perform an activity with onion bulb. Aim to observe cells in onion peel. Materials required: microscope, slide, onion bulb, water, blade. Let's see procedure. Peel on onion and cut out a small fleshy portion from the onion bulb. Break this into two small parts and try to separate in thin, delicate, transparent membrane from it. Carefully place membrane on clean glass slide. Add a drop of water on slide and cover it with cowrie slip. Observe under microscope. Let's perform an activity. Let's see the materials required: onion, water, dropper, blade, slide. Cut out a small fleshy portion. Try to separate a thin, delicate, transparent membrane. Here is the membrane. Take a slide and put it on it. Add a drop of water. Here is cover slip. Place this cover slip on slide. Press gently. Observe this under microscope. Under microscope, we observed rectangular shaped cells. The cells are covered by cell wall. Here are the cells seen under the microscope. Cell wall.
Middle nucleus.
Middle nucleus. Cells. Cells in onion bulb. At last, result of the activity is we observed cell in onion bulb. Thank you for watching. Let's meet in another activity. Hello friends, activity number 2. Aim to observe cells in human cheek. Materials required: microscope, slide, water, covered slip, dropper. Let's see the procedure. Wash your mouth cleanly. Take scraper and scrap the inner side of your cheek. Collect the material and put it on the slide. Add a drop of water. Cover with cover slip. Observe under microscope. Let's see the material: water, dropper, cover slip, scraper, slide. Friends, take a scraper. Friends, now scrap the inner side of your mouth. Now take a clean slide and put the material on it. Now add a drop of water.
Very good.
Cover it with cover slip. Press gently. Observe under microscope. Observation: we observed irregular shaped cells under microscope. Friends, we observe this structure: cell, nucleus, cell membrane, cells in human cheek. Result: we observed human cheek cell. Thank you friends. Let's meet with another activity. Bye bye.
Okay students, so the next part after nucleus is diversity in cell. So what's the meaning of diversity? Diversity is simply, we can call it as variations. What is that? It is called as variations. Okay, now once tell me one thing, do all the cells are in same shape and size? No. If you see a small organism like rat, or if you take a huge animal, and that is for an example elephant. Now tell me one thing, do the cells which are present in rat and the cells which are present in animal, are they same? No, they are not. Okay, if for an example, if you take, for an example I am telling you, imagine that a rat have 100 cells, imagine, whereas in the case of here if it is 100 cell, elephant it may have some thousands of cells. That's it. Cell is a cell. Okay, cell will be microscopic in size, it will be very small, microscopic in size, we cannot see with the naked eye. Either it may be a elephant cell or it may be a rat cell. Cells are microscopic in nature, and the cells which are present in any organism, they are not similar, they are not round, they are not completely hexagonal, they are not, all the organism, they don't have the similar kind of cells. There are different cells which are seen in different organisms. So all that differences make diversity in cells. Differences are called as variations. Okay, so the diversity of the cells are nothing but variations which are shown by an organism. Variations which are shown in cells are called as diversity in cell. Whereas if you come to cell is divided into unicellular and multicellular organism. So cell is divided, organism is divided into unicellular and multicellular. So already I explained about this, but for a quick review I will explain again that unicellular, the word itself, uni is nothing but one. What is the meaning of uni? Uni is nothing but one. Cellular is nothing but the organism which contains cell. Okay, so how many cells are present in such kind of unicellular organism? Only one cell throughout its life. Throughout its life, the organism which have only a single cell. In the case of multicellular organism, multi is nothing but many. Multi is nothing but many. So these organisms who have many cells in its lifetime. In its lifetime, how many cells do they have? They have many cells. They have many cells. Okay. So the example for unicellular organisms is amoeba. The example, one of the example if you consider here, it is amoeba, Echinomonas, Euglena, Paramecium, Escherichia coli, E. coli, we call it as E. coli. Okay, so many, you can give many examples under unicellular organisms, but for a simple idea in this level, if you have an example of amoeba, Paramecium, Euglena, so that's more than enough. Whereas coming to multicellular organism is nothing but the organism which have many cells in it, the organism which is made up of many cells, and the example for that is plant, animal, even some microorganisms also like fungi. Fungi is a microorganism, but even though it comes under multicellular organisms, right? So this is a organism or cell which is divided into two types. Here, if you take unicellular organism, it have a single cell, right? In the case of multicellular organism, so the diversity which is seen by multicellular organism, if you take an example of human body, we are multicellular organisms, yeah, we are the multi-celled organism. We have many cells in our body. Start from our head till our toe, we have many number of cells. Okay, the cells which are present in my eye, the cells which are present on my skin, the cells which are present in my stomach, the cells which are present in my hand. So all these muscles, nerves, so we have many number of cells in our body. So and the many number are not in same size, they are in different shapes. If you take an example of nerve cell, it is like something like branched. It is something like branched. If you take blood cells are again, it is round in size, shape. Okay, so in this way we will find different variations in even a human body. Okay, so this is about diversity in cell. So these are the examples which is included in the textbook. Once if you observe, so this is a structure of Echinomonas. It is unicellular organism. It is found in fresh water, found in marine water, even it is found in stagnant water where the water it won't move, like pond, okay, canal. So there also you will found this Echinomonas. Okay, it is a unicellular organism. It is it is called as green algae. Green algae. The algae wherever you are finding here and there in the water, upon the water you will find the green patches like. So that is Chlamydomonas. It is green algae. Okay, and the name of the green algae is Chlamydomonas. Next to that, it is a structure of amoeba. What is that? It is a structure of amoeba. Amoeba is an organism which is very special. A reason behind that is it is included under unicellular. It have a single cell. It have a single cell in its body, whereas in the case of this organism, here the speciality of the organism is it doesn't have a particular shape. I have drawn in this way. You can draw something like this. Whatever, however you want to draw, you can draw the structure of amoeba. The reason behind that is it have a ability to change its shape. Okay, how it will be changing its shape? By this pseudopodia. Imagine, now I will say how the amoeba moves. Amoeba doesn't have any foot. Amoeba doesn't have any foot. Yeah, even though without any foot it will be moving here and there inside the water. Where you will find this amoeba? Amoeba is a unicellular and it is found in water. Mostly it is found in outside, but whereas some cases it is found in the human body also, and it may cause some illness in the human body. So how it will move? Imagine that it is a food particle which is present inside the water, and here it is amoeba. This is amoeba, and this is a middle part which I have drawn here, this middle part, and the projections which I have kept here with the red color. Imagine that these are pseudopodia. Okay, these are pseudopodia. So here is the food material, right? So this algae, amoeba, what it will do? It will be coming like this, and then it will be holding, or it will be engulfing the particle, engulfing the food material inside its body, and it will release some chemicals on it, and then it get digested. Okay. So here it doesn't have any mouth. Amoeba doesn't have any mouth, but the body itself move in such a manner to engulf the food material. It will be completely engulfing the food material, and then it will be digesting the food material inside the body of an organism. Okay, so that is the speciality of amoeba. Okay, amoeba is a unicellular organism. It is found in water. Next, coming to the last one, that is E. coli. E. coli is nothing but Escherichia coli. It is a bacteria. It is also a unicellular, or else even it is found in colonies. Okay, now where do you find this E. coli? E. coli mostly it is found in our intestine. Where it is found? It is found in our intestine. It is not a harmful microorganism. Don't be tense that bacteria it is found, it is present in your intestine now. It is present in our intestine, but it is not a harmful bacteria. It is useful for our digestion. Okay, but in some cases, some of the E. coli species, some of the E. coli strains, they may cause food poisoning also. Okay, so when we are discussing in the next chapter, story of microorganism, we will discuss about all these organisms in clear. Okay, so the next topic which is there after diversity of living organisms is observing cells in a leaf. So even you can take an example of leaf, or even you can take an example of stem. Okay, here something which is given to our stem. Okay, so if you take an example of a stem, you take a stem part, cut it in a transverse way. If it is a stem, cut in a transverse way. So this horizontal cutting is called as transverse section. So if you take a stem, cut it into a transverse way, and then you will find this type of structure. Okay, so if you see the name here, transverse section of dicot stem. So for seed is of two types. You know seed, what the seed is? Inside the fruit you will find seeds, right? All the flowering plant have seed, and that all the flowering plant have fruit, and inside the fruit you will have seed, and this seed is of two types. Okay, one is monocotyledons, monocot, and the second one is dicotyledon or dicot. Take mango seed, sow in the soil, and that will be producing 2 leaves at a time. Okay, so when it is germinating, it will produce 2 leaves. You can take another example of tamarind also. When you sow in the soil, so it will produce 2 leaves. Okay, it will produce 2 leaves. So that 2 leaves, it indicates that it is a dicot stem. Okay, dicot stem or dicot seed. Dicot is nothing but dicotyledon. Di is nothing but 2. It have 2 cotyledons. So any flowering plant like mango, tamarind, you can take an example of guava. you can take an example of apple, whatever flowering plant. If you take those flowering plants in a biological term, we call it as angiosperms. Okay, why it is called as angiosperm and what are the general characteristics of angiosperms we will discuss later. But whereas you have to understand that flowering plants are also called as angiosperms, that has a pair of leaves. It have a pair of leaves. The very good example for that is mango. I hope you have observed it. Okay, mango, when you sow in the soil, it will produce 2 leaves. It will germinate, it will be producing 2 leaves after germination. So imagine this is a seed and these are 2 cotyledons. These are 2 cotyledons and it will produce 2 leaves here. And that 2 leaves, it will be growing as a tall tree or plant. So that cotyledons in the embryo or the seed. Embryo mean it is present inside the seed. Okay, so this is something about transverse section of the dicot stem. When you come to the parts which are present in a stem, so you can see here epidermal hair. It is the outermost layer. That epidermal, see these small hair like structures are called as epidermal hair. Beneath the epidermal hair you find epidermis. Epidermis, again you have hypodermis, cortex, endodermis. We will discuss all when we are discussing in plant tissue. Okay, in plant tissue we will discuss all these parts, but now you have to know at least these names, that is epidermis, hypodermis, cortex and endodermis. These are the inner layer of dermal tissue. Okay, so the outermost layer of a plant cell is called as dermal tissue. Dermal tissue, again it is divided into epidermis, hypodermis, cortex and endodermis. Next to that you will find this green color part. This green color rounded bundles, and we call these as vesicular bundle, and these vesicular bundles are helpful for transport of food and water from up to down and down to up. Water, I mean if you take an example of roots, roots will absorb water and minerals from the soil, and leaf, which is in the shoot system upward, right. So what it will do, roots will absorb water from the earth, it will send to the upper part of the plant, whereas in the case of leaves, it will prepare food material. As it contains chloroplast in it, it will prepare its food material and it will send to the remaining parts of the plant body. So in this way the transport is done by vesicular bundle. This green color part, it contains xylem and phloem in it, and that xylem and phloem is helpful for transport of food and water. Next to that, here in the middle of vesicular bundle you will find ground tissue like parenchyma, collenchyma, sclerenchyma, storage tissues, erenchyma. There are different types of cells which are present in ground tissue. We will discuss about each and every tissue in the next class. Next, in the middle you will have a dense body, and this is called as pit. Okay, so this is a complete transverse section of a dicot stem. Up to now we saw diversity in plant cells or diversity in microorganisms, whereas in the case of now we are going to say diversity in living organisms and especially animals. So in animals we will find these many, not only these what I have written on the board, we have many number of different sizes and shapes of cells in our body. So for an example, if you take muscle cell, muscles are useful for movement of our body. Okay, so movement of hands, movement of legs, limbs. So we want some muscles. So that muscles are of 2 types. Those are 2 types, voluntary and involuntary. So whereas in the case of here in the textbook it is mentioned smooth muscle cell. Smooth muscle cell is an involuntary muscle. Okay, it is an involuntary. The shape of involuntary muscle or smooth muscle is this. Next to that it is bone cell. If you see our body have a skeleton and our body have a particular shape or size, and that is because of bones, arrangement of bones, right. So that bones here, this bone cell have different cells again like osteocytes, osteoblasts, bone lining cells. We have many number of cells in the bone cell, whereas this is one of the cell and it is like this. Okay, so this contain nucleus, this also contain nucleus in it. Nucleus, if it contain nucleus, then it must be clear that nucleus have a property to divide. Okay, so these cells are continuously dividing in the body, whereas in the case of here we have one more cell and which is branched like a tree, and this is called as nerve cell. What is that? So here you will find a central body called axon. To the axon we will see some hair-like structures which are attached, and these are called as dendrites, and a longitudinal tube-like structure, and at last you will find nerve terminals. Okay, so the function of nerve cell is to carry the information from brain to different body parts. Now imagine I have a marker and I want to hold it with my hand. So the nerves which are present in the brain should give a signal to my hand to hold this. Okay, so what the brain will give a signal through this neurons, the information will be transferred from one neuron to other. Next to that, if you see blood cells, blood is in red in color, right? Most of us know that. If there is any cut, blood oozes out, and the blood which is coming out or oozes out, it is in which color? Red color. So mostly we will know that blood is in red color, so the cells which are present in it are only red. But no, blood have three types of cells, that is red blood cells, white blood cells and platelets. Okay, red blood cells, white blood cells and platelets. Whereas in the case of red blood cells are also called as erythrocytes. What it is called? It is also called as erythrocytes, whereas white blood cells are also called as leukocytes. Leukocytes. You can write C or K there. Okay, it is called blood cells, human blood cells are divided into red blood cells, white blood cells and platelets. So red blood cells are also called as erythrocytes, red blood cells are leukocytes. Okay, whereas in the case of red blood cells, the name itself indicates that it is red in color. Okay, it is red in color, whereas in the case of white blood cells, they are white in color or colorless. Okay, where this might be in spindle shape or circular shape. What is it? Red blood cells. Then what is the function of red blood cells? Red blood cells are helpful in transport of oxygen in the body. Okay, so these red blood cells are formed from hematopoietic cells, and these hematopoietic cells mean these red blood cells have a pigment called the hemoglobin, and this hemoglobin have a property to carry the oxygen which we are inhaling. When we are inhaling, inspiration process, in the inspiration process we will take oxygen in. So that oxygen through our lungs, it will enter into blood. Okay, so from the blood it will transport throughout the body, that is by a red blood cells. So the function of red blood cells is carrying the oxygen molecule throughout the body. Next to that, if you come to white blood cells are also called as leukocytes, and again it is of two types, granulocytes and agranulocytes. Okay, granulocytes and agranulocytes. Granulocytes mean it have granules in it, whereas in the case of agranulocytes we don't find any granules. Then what is the function of white blood cells? So up to now I said red blood cells will carry oxygen molecule, whereas in the case of white blood cells are also called as police cell. They are helpful for protection, they are helpful for immune system, to build up immunity. There must be a cells, and that is called white blood cells. So here if you see basophils, granulocytes are divided into three types: basophils, neutrophils, eosinophils. Next, agranulocytes are divided into monocytes and lymphocytes. When we come up to now, I said that all the cells which I have written on the board, which I have drawn on the board, are not big in size, and they are not seen by naked eye. They are very, very small in size, they are microscopic. So even if it is microscopic, then how we are identifying it? And that is because of microscope, because of the invention of microscope. For our easy calculation, it will be useful in our laboratory to measure the size of an organism. Once if you look at here, 1 meter is equal to 100 centimeters. We knew it. Okay, this 100 centimeter is in a short form, it is called as cm. Next, 1 centimeter is equal to 10 millimeters. Okay, next 1 millimeter is equal to 1000 micrometers. What it is? 1000 micrometers. Micro is written like this. Next, 1 micrometer is equal to 1000 nanometers.